The overall goal of this article is to reconstruct a reliable phylogenetic tree from DNA or protein sequences. This is accomplished by first identifying similar sequences using BLAST programs at NCBI. The second step is to align similar sequences. Next, the best fit model of evolution is determined from the alignment. The final step is to infer the phylogenetic relationship from the aligned sequences. Ultimately, the step-by-step pipeline is used to show how users can go from sequence data to reliable phylogenies.
This method can help answer key questions in diverse fields by inferring identity and function to novel sequences.
To use the online version of the Basic Local Alignment Search Tool, or BLAST, navigate to the National Center for Biotechnology Information, or NCBI's BLAST web server. Click on the appropriate BLAST program. Input a FASTA-formatted text sequence, such as the one shown here, into the query box. Click the appropriate BLAST program for use in the search, and then click BLAST. The output is in HTML format by default and displays the most similar sequences to the input text sequence. The next section covers using a local BLAST executable on Windows. Mac users can skip to the following section, called BLAST Local Executables for Macs.